feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the last show of June, the National University of Singapore Yong Lin School of Medicine's Healthy Longevity webinar. I want to remind you before we start to use the Q&A function to send your questions. The questions the last few weeks have been really good, so I'm expecting good ones tonight too. We have Professor Gunter Lepperdinger tonight with us, and I'll be introducing him in a little bit. Prior to doing that, though, I want to introduce Fan Ben Chiang, who's a UROPS undergraduate student at the Center for Healthy Longevity. She will present on her project, Establishing an In Vitro Senescence Model for Studying and Targeting Aging Hallmarks.
Thank you for the kind introduction. As mentioned, my name is Bin Tian, and I am an undergraduate student from the Sorrentino Lab at the National University of Singapore. Today, I will be presenting on our project about establishing an in vitro senescence model for studying and targeting aging hallmarks. Aging is a process that is characterized by different cellular hallmarks, one of which is cellular senescence, a state where cells stop dividing permanently. Some other features of cellular senescence include being resistant to apoptosis and displaying senescence-associated secretory phenotype, also known as SASP. While senescence cells were initially shown to serve beneficial roles in tumor suppression, they can also induce inflammation by secreting pro-inflammatory substances. This can contribute to chronic inflammation and eventually drive the process of aging. The studies on SASP has laid the groundwork for targeted drugs such as senolytics that are designed to selectively get rid of senescent cells from the body. Research has shown that the combination of different senolytics are able to reduce health issues such as diabetes and Alzheimer's disease in preclinical models. Aging is also highly associated with the regulations of NAD levels in our body. As we age, NAD is being depleted by various cellular processes in our body. This decline in NAD levels can affect the functions of mitochondrial enzymes and significantly impact cellular metabolism. Hence, boosting NAD levels in our body by taking supplements can improve mitochondrial function and delay various aspects of the aging process, including cellular senescence. Our lab is also interested in studying the association of cellular senescence with other hallmarks of aging that are linked to NAD metabolism and mitochondrial dysfunction. By establishing an in vitro senescence model, we will then be able to study how these factors affect senescence and the aging processes. Our experiment aims to construct an in vitro senescence model with mouse embryonic fibroblasts by treating them with drugs that disrupt the cell cycles to induce senescence in these cells. Mouse embryonic fibroblasts are chosen as the model organism as it comes from a finite cell line and shares similar senescence biomarkers as human fibroblasts, which makes it a relevant model for understanding human cellular senescence. When the cells undergo senescence, they display certain characteristics. Besides being enlarged and having an irregular shape, they also display an increase in beta-galactosidase activity. One way to detect beta-galactosidase activity in the cells is by staining them with XGAL, a substance that produces a blue product upon reacting with beta-galactosidase, as shown on the picture on the right. Here are some microscopic images of beta-galactosidase staining in MAS cells. The cells were treated with 3 different types of senescence-inducing drugs, namely bleomycin, peroxyblate, and doxorubicin. As indicated by the red boxes, the greenish-blue staining that were observed shows presence of beta-galactosidase activity. These stainings can provide a brief overview of the treatment groups that are effective in inducing senescence in the cells. However, as it is difficult to compare the relative intensity of the stainings by the naked eye, other methods are required to accurately quantify beta-galactosidase activity in the cells. Hence, a quantitative fluorescence assay was also performed. In this assay, a reagent called 4-MUG is added to the cells. Beta-galactosidase hydrolyzes this reagent to produce a fluorescence compound called 4-MU. We can then measure the intensity of the fluorescence produced to quantify the amount of beta-galactosidase activity present in the samples. Results shown here indicates that most treatment groups shows a significant increase in the detection of fluorescence compared to the control, which indicates that cells in these treatment groups shows a significant increase in beta-galactosidase activity after being treated with senescence-inducing drugs. Besides investigating beta-galactosidase activity in senescence conditions, we also need to look into other senescence-related biomarkers, such as the protein P16 and P21, to further validate the senescence model. With its establishment, we can then utilize the model further to investigate how senescence is related to other aging hallmarks, such as mitochondrial dysfunction and the loss of proteostasis. Thank you for your kind attention.
Thank you, and we'll look forward to hearing some of the results of that project. Gunter Lepperdinger is very well traveled and well studied. He was a biochemist at University of Vienna, a developmental biologist at NIH Bethesda, a biogerontologist in the Austrian Academy of Sciences, and currently is in the University of Salzburg in the cell biology and genetics. The starting point for his research was really on embryonic development, and that transition to the using stem cells to try to understand more about developmental processes. Currently, he and his team are designing and manufacturing bioreactors in which the human bone marrow is reconstructed, and thus the natural aging process and also pathological processes thereof can be made comprehensible. Tonight, he'll be talking a little bit about that with this topic, deconstructing and reconstructing bone. But I also, before he starts, I want to mention one thing. This is the first show that I think we've done where Gunter's actually using my office. So I just want you to know, Gunter, that I know what every piece of item in that office is. So if you steal anything, I'm going to know about it. Okay.
Thanks, Brian, for using your office. Thanks for inviting me. So in this case, I should start with good evening, Singapore, but also good afternoon, Salzburg, or good morning elsewhere in the world. Thanks for having me here. Yeah, Brian introduced you to my topic already, which is on bone, and we had the chance to actually look into that from the perspective of healthy longevity in a way that we thought it would be cool to address the question, what's in bone, in a way that we know more. But at least at that time when we started, this was 20 years ago, the technology was not good enough to look into bone at a cellular level. So we started by arguing we have to deconstruct the bone first thing. And later on, we had the chances to get into touch with technology that's evolving to reconstruct the bone now. And I'll tell you a bit about this story and what we could actually achieve. So the idea of looking into bone biology and aging and pathology is a very simple one. If you look into young bone, you'd find with the actual texture of the cortical bone, little cavities where cells are residing, osteocytes, but you'd also find a lot of vessels there and nerves. And sure enough, this bone structure changes over time, and that became kind of interesting for us, not also with respect to the cortical bone, but also to the bone, which is the bone marrow, sorry, which is adjacent to the inner side of this bone. And we thought, why not looking into that more closely, as also most of you are aware of, bone marrow is not only having bone marrow on its own, but a lot of stem cell niches. And that became interesting in a way that we thought we have to work on humans, although sure enough, the model system of a human is not as easy comprehended that of a rodent. But on the other hand, a short-lived creature like rodents might not be comparable to a long-lived situation, as we know that from humans. So we started to deconstruct the bone marrow, as we were fortunate enough to have collaboration partners at Innsbruck clinics who were craniofacial surgeons, and they often take bone and transplant it into a facial region to reconstruct the face or maxilla or mandible. And the remnants of this bone, we were able to get and isolate cells from. So the first idea was to get some idea what type of cells we can isolate in order to look whether cells derived from old bone or elder people show something like we know from fibroblasts, and whether this type of cells, which we were after, the mesenchymal stem cells at that time, they were called stem cells or Kaplan cells, are as old as the individuals we derived them from. So we invested a bit of time, to isolate, to come up to standardized isolation protocols, and also to find markers, defined markers, and specify assays, which we could actually use to look into the question more closely, whether the bone cells which we have now at hand show something like a reliable measure of the individual's or donor's age. And by doing so, there were a couple of assays also revolving around the question whether these cells are real stem cells, whether they are multipotent, or what they can be actually used for in terms of tissue engineering. We posed a simple question whether these cells are comparing to the donor's age. And by collecting many samples and now doing something like an extrapolation and with a regression analysis, we could find, at least for us, it was good enough to say that the extrapolation would end with cells being that you're running out of cells or these stem cells only if you would end up with the age of 300. So that said, we thought, okay, we have other assays as well. And the most tedious one, which we have in our field, is replicative senescence. And the show before already showed you that replicative senescence is kind of a model system many people are interested in. Here we just used it to accumulate population doublings, which we wanted to count in order to show whether the cells already underwent aging, replicative aging in vivo. And now if you do so, you end up with a number of, say, 100 years where you might still have enough cells, but those cells that are around are not good enough to replicate or to proliferate anymore. Therefore, one could argue this is either stem cell exhaustion or something that one might use as a clock on its own, like an aging clock. So our other measures were, could we actually somehow rejuvenate or give the cells relief. And we found out that oxygen is interesting, but not only in order to enhance their proliferation capacity, but also in order to manipulate the ways the cells are differentiating. So we found out that at 3% in vitro oxygen condition, the cells are tending to proliferate much better. But if you put them in ambient oxygen condition, which in the lab is 20%, but which you never would find in the body, these cells are more prone to actually undergo differentiation. So that was interesting. And also what you see down here with the oxygen condition shifting from 3 to 20 would tell you that under 20% ambient in vivo conditions never found, but 3% you can easily think of having that in the bone marrow. These cells are producing just much more colonies, which is a good measure for stemness. So we argued that changing the milieu is very much something that cells would sense and would appreciate, would actually use to guide their potentiality towards differentiation. We also looked at that time, the inflammaging hypothesis came up, whether these cells would also be insulted or offended by pro-inflammatory cytokines, what they actually are. So again, here they're hampered in the differentiation capacity, while sure enough, if you want to differentiate these cells, this is kind of a guiding signal, as wound healing or regeneration often also uses transient pro-inflammatory cytokines being around in a local manner, albeit inflammation would be systemic inflammation. We could show that if that is systemically given, the stem cells are kind of irritated with respect to their differentiation potential, now being not as prone to differentiate along the osteogenic lineage, but more towards the adipogenic lineage, which coined the term of adipogenic switch for the field. Now, we concluded at that time that we think we could call it we are as old as our stem cells. So the in vivo situation was reflected also by the cells when we deconstructed the bone and isolated the bone marrow cells. Now, sure enough, we couldn't actually tell a lot about the stem cell niches, because when extracting bone marrow or isolating bone marrow cells, we threw away the niches. So our next step into this unraveling this question was, how could we actually reconstruct the human bone marrow, which we started by finding out means and measures to get into 3D biology. So most cell biologists working in 2D, meaning that the cells are cultured on plastic surfaces. And now we thought about methods where we can actually take away the plastic surfaces and let the cells attach to each other and now form these little nice creatures, where we supposedly would also find stem cell niches. So the idea is that you could aggregate these cells, and this is not a new technology. Embryologists did that already 100 years ago. So we adopted that technology and looked whether we can again find some nice assays that would illustrate what's going on. And still the gold standard in this sense is histology, which is tedious, but that's the life of a scientist anyway. So that shows you just a couple of assays we can do. We can use again the plastic surfaces and put these little aggregates on top of the plastic surfaces and let the cells spread. So we do have also something like assays that are combining what we already know about the cells when we isolate them. On top, you see this aggregation. We come back to that later. That could be easily followed in time lapse. We can also use a lot of fluorescent dyes, don't actually expect that the colorful pictures are very, very in focus, because here we do have hundreds of micrometers which the cells are now as an aggregate are, and sure enough, microscopes are not able to put that in focus. You could think of laser scanning microscopy, but there is also constraint to that. So again, what the next picture shows you is, if you're working with cell aggregation and you use different cell types, you often find that the cells, when they aggregate together, that these cells are starting to have a necrotic core. So the first actual problem was to solve whether these cells, which we are using here as a surrogate model for the mesenchymal stromal cells, which is osteoblast cells derived from a human fetus, that they, once they are aggregated, are actually not forming in a necrotic core, albeit these little cell piles could actually grow for many, many weeks and months. They are growing and getting larger and larger. They are not necrotic in their core. But what we found is that they actually underwent something like a spontaneous osteogenesis, which was interesting for us. So we supposed that we have now a good model of, that undergoes self-organization, and that we could just follow it like a kin or similar to a human embryo, which it actually not is. It's maybe also not an embryoid body, but comparable to this idea. Now, as this needs certain new technology and instruments, we actually built up a little workshop where we build now instruments where we can actually come up with tools that help us to study this aggregates and the formation of larger tissues, which I think one could now call microtissues. So on the right side, you see the merging of three of these aggregates, which we formed separately and then put together. And what you see here is, for instance, which is called GM and OM, two aggregates which fuse together or merge or grow together. And this OM was treated differently than the GM. And you see in histology that they actually look differently. And when you grow them further on, they stay different with respect to this histological staining, meaning that there is more of collagen within the OM and less in the GM, and this is surrounded by cells that are heavily proliferating, so that the tissue is actually growing. And by showing you this triple staining, meaning that you can use this aggregates similar to little building blocks or bricks, you can now build certain diverse tissues from starting from different building blocks. So this is the kind of idea we are following right now up. But also possibly is that you either co-aggregate different other cell types, or after aggregation of certain basic cell types like mesenchymal stromal cells, you could add other cell types to that aggregate. For instance, in this case, the THP1 cells are monocyte precursors. And when you add them on the outside of this little aggregate, they will actually migrate into the core region where, for instance, endothelial cells added to this aggregates, they stick on the surface. So here again, we could do some basic cell biological research on how cells are interacting in 3D and organize themselves not only with respect to histological characters, but maybe also with respect to undergoing now developmental pathways that tell or guide them towards a functional tissue. So that said, we think we're right now there that we can form or build cell environments which we could consider being tissue primordia that actually also contain stem cell niches. Being confident about that, we are currently working on hematopoietic precursor cells, which we add to these cell aggregates, and we are looking for certainly enough. Not the entire story would be also to have some vascularized microtissues there for the hematopoietic niche, which is a bony niche, but also a vascular niche. There's two niches for the for hematopoiesis. You could also now put immune competent cells in there and ask whether this kind of bone marrow type of aggregate serves as immune niches. So that's also interesting for us. In order to form more structured, convenient niches, we also use cellulose hydrate, which we could plasma treatment more and more. And you'd see if you plasma treat, these cells are using this type of biomaterial surface in a way known for normal cell culture. But if you just treat it a bit, these aggregates just sticking there. So again, you could here now use cells in the surrounding like feeder cells that are integrating or using a bit more of plasma treatment so that spreads and you find more stratified layers of cell tissues. We are also interested in using bony tissue, and here we found out that, for instance, polyethylene glycol 700 in size could be mixed together with hydroxyapatite. And if you put that into the vicinity of these cells, they ought to react like having here now an artificial bony niche, as we could also use little particles of hydroxyapatite and co-aggregate. And this is without hydroxyapatite, and this is with hydroxyapatite, now using the endogenous alkaline phosphatase as a tracer of showing changed differentiation capacity. So they're reacting and sensing their micromilia, which we can now easily manipulate. And in order to form something like a vascularized tissue, we now build a device which is actually kind of a heart. So we have here a plastic bag which holds medium, and we have little valves, and here we can push in the media through this flow chamber in an oscillating manner, like a pulse. Every second we push some media in, and this is pushed into the bag. And if the pump, which is a syringe pump, runs out of media, it pulls back the media into the pump and would start again pulsating into that. So here we can do a bit of live cell microscopy in this kind of pulsing chamber. And what we see is, if we do so in this cellulose hydrate system, the cells are not just sticking to the surface and are contact inhibited once they are turning confluent. Now they start to actually grow on top of each other, now having more and more layers for forming microtissues. So here we are pretty confident to say that we are now up to building new microtissue surrogate models of human bone marrow in this kind of biochip, organ on a chip format. So if I should carry on. Just tell me, Brian, if there is some time left. I'll just show
Yeah, you have a few minutes. Take your time.
Okay, good. So what you see here is a developing chicken embryo, and this chorion allantois membrane is actually there to cover the entire chicken yolk, and the embryo, which is growing vastly, needs the vascularized chorion allantois membrane to actually digest or ingest the yolk in order to grow the embryo. And this is kind of a nice model system, which is again around for ages, which can be used to put certain stuff on top of the chorion allantois membrane, and normally it's taken into toxicology assays. And here we use that for putting on little reactors, which we use to stick on there with silicone rings. And we could show that here, when we put cells in this little chamber, we do have now cells that aggregate, that grow, and that interact with the growing vascular system. So this is also something that we could follow up in case we're very eager to find vascularized tissue, albeit this is now a chicken human system. The other system which we're trying to get working is human endothelial cells together with our stromal tissue. So our conclusions are that we are able to work with bone marrow cells in order to form stable aggregates. We can grow them in flow chambers. We're confident that these things, once they can be co-cultured with other cells in 3D, can be used to be grown and manipulated under near physiological circumstances and conditions. So the perspectives is to learn more about co-aggregation of aged cells in this system, to using microvascularization of microtissues, and the molding of hematopoietic and immune competent cells. And that said, we also use that kind of technology to proceed into in vitro, from in vitro to in vivo. And here we are just using a technology which is called impedance measurement. And without going into details, the idea is here that you're using electric conductivity, which is different from blood to bone, in order to measure whether there is bone forming, and just showing you that once there is cells that are forming bone, like osteo and not adipo, you'd see different measures that you can easily sense with this kind of technology. So we use this interdigitated electrode sensors and build that in a way that we can actually put that into a bone defect, screwing that in, and this is what you see in X-ray. And again, here that shows that this kind of sensory method can be actually used to monitor inline whether something is forming towards bone, or this was in the neck, in the skin tissue, whether the baseline stays normal. We call that now a chip in an organ, so that we could actually also have some non-invasive, sure enough, this is invasive, but invasive sensory technology that would allow us to monitor what's going on underneath this sensor surface. And here we do have something that actually ossifies, which I already showed you. There is something that ossifies a lot, and here we would have something that ossifies less. And that said, we are up to also introducing that as a new technology, or presenting that as a new technology to orthopedics, now having something in mind that you could code, for instance, osteoprothesis, endoprosthesis, and using that in clinical future to actually control the fixation and the ingrowth of osteoprosthesis in patients. So that said, wrapping up, our journey started from 1D over 2D to 3D and ends up now, or should end up, in vivo chip systems. Age well.
Thank you. It's a really interesting talk. I think one of the things that strikes me, and I'm hoping my work is in a different way going in this direction, is we started out asking very, very basic questions in science, and it must be gratifying to see that you're really moving rapidly toward applications that could really impact people's lives.
Thank you.
I wonder if we could start somewhere very basic, because we haven't talked that much about MSCs, and so some people in the audience they've heard. I'm sure most people have heard that, but MSCs can be derived from different places: bone marrow, umbilical cord. There's adipose-derived stem cells, which have similarity. Maybe you can just comment briefly on the different kinds of MSCs and how similar they are to each other.
Yeah, we tended to say in former times that there is a fibroblast, and many people worked on fibroblast aging. But if you look into the nature of fibroblasts, you might end up with, say, 200 different types of fibroblasts in your body. And this is also true for mesenchymal stem cells or stromal cells that taken from fat or from bone or from skin, there tend to be a difference. We think that the most multipotent mesenchymal stromal cells are found in bone marrow when it comes to the adult situation, and you already named it. If it's the umbilical cord where you derive them from, they're kind of fetal. You'd also find them very broadly in their determined in that differentiation potential, and they're also eagerly growing because they're still young.
So, well, first of all, I'm happy that they have this technology that they can take MSCs from bone and put them in repairing bone, because that's what happened to me when my femur was destroyed. They took bone from my hip and along with a lot of metal, grew back my femur. And that's relatively old technology. That accident was a long time ago now. So, yeah, that's another thing that struck me during your talk, is that this idea of growing organoids we think of as very new, but this kind of technology in many ways existed a long time ago, at least for bone, it sounds like.
Yeah, going back into the embryological literature, it would be Holtfretter in the 30s who actually started doing something like disintegration of embryos. They found out that if you just take away the calcium and the magnesium, that they fall apart, because it's this CAMs, the cell adhesion molecules, that are relying on calcium and magnesium. And sure enough, you could play around like that, aggregating, aggregating the cells, and find out what you should actually put into this cell suspension, that you end up with something that actually grows or can regrow to a certain extent in a fashion that it resembles tissue. So that's a good model system on the one side. But on the other side, you could also think of what surgeons are doing, that you just put something that you take from your body at the spot, put it together, and reintroduce it into your body. And for instance, here they're using a lot of fibrin-derived stuff, like this platelet-derived stuff, and that works brilliant. It's really good in order to remodel your traumatic tissue that is harmed. So there's a lot of things there that are actually working in the clinics, and I guess one also should to talk to these guys. They are inventing a lot of methods which we are not aware of in the lab, and talking together, talking about these points and issues actually nurture both sides.
You mentioned that you showed data that the bone marrow derived MSC senesce, they senesce earlier from older individuals. What do we know about the properties of senescence of those cells? How would that compare to, say, a fibroblast if you look at, say, secretory profiles or other things? Or is the SAF relevant for these bone cells?
I don't know so much about the details, but I know from again clinical doctors who stepped into aging research, and they looked, for instance, beta gal and what they express on a very superficial level, that you'd find many senescent cells. For instance, in femur, when they take it out and they just take the cells and they want to let them proliferate again. We actually also had kind of a morbid idea, taking bone marrow from corpses. And when you do that with very old people, because they are more likely to die and they are more likely to be available, you'd end up with very short-lived cells. So they senesce, and whether it's different to another type of senescence, I can't really tell you in detail.
Have people tried reprogramming these cells and then differentiating them back into MSCs? And
Yep.
in this case, does the oldness is that retained through reprogramming or not?
Exactly, that's the case. So that has been done, and also could reflect what is done in other systems, I guess. So the epigenetic marks are still there, and how they can be changed is still under investigation, I would say. We also tried a bit. We have a couple of epigenetic markers which tell us how old they might be and whether we can use them. And we have, for instance, used one senolytic drug like visitin to show whether these marks are different than, but we didn't follow it up to the end, to that end, to show that this is a real rejuvenation, that any type of assay would give us a good clue, for instance, stemness or proliferation capacity or the differentiation capacity, that actually this was a true rejuvenation.
So I think these biomaterials are fascinating, and I think you're trying to recreate tissue-like material so you can study them, and now their applications a little bit. A lot of people are probably thinking, this is on the way toward recreating a bone. I mean, how far technologically are we away from generating a whole, for instance, a bone? I mean, we talk about organ replacement and tissue replacement. Where are we in the bone set context?
At least this is my impression. We don't make a lot of progress.
Yeah,
There's a lot of
yeah.
bone substitution material around in the clinics that actually works well, and you have already named it. Your own autologous bone is much better than anything else, and the point actually here is that they don't take out just bone; they always take some vessels with it. So has to has this anastomosis. Otherwise, it undergoes necrosis in a few hours, I would say, and this is how it actually works. And therefore, we are so interested in vascularization of these microtissues.
I see. That's kind of a next step to preserve their function longer.
Yeah.
I missed, how do you, in the context of the biomaterials or even in the organoids, how do you induce an aging-like phenotype? Maybe you mentioned this, but is that by adding inflammatory factors or trying to drive accelerating aging through other mechanisms? we've thought about this a little bit in the context of astrocytes and neurons. So I'm wondering how broadly you've looked at ways you could accelerate aging in these cells.
What we are doing is two things. We are using a lot of cells, and we let them proliferate in order to amplify, because cell aggregation needs millions of cells. So they do undergo replicative aging anyway. On the other hand, we're using very, very little amounts of TNF alpha, meaning this is in the nanograms per mil range. And that also works in a way that they show phenotypes such as inflammation phenotypes. Yeah.
Interesting. Maybe I'll bring Shara in. She's one of our regulars on the show. So can you tell us what are the best questions from the audience today?
Okay. Thank you, Prof. Brian and Prof. Gunter, for the discussion. So there are actually quite a couple of interesting questions from the audience, and I think perhaps we start with the more general ones before we move into the technical questions. So from the general questions, one of the audience asked, what are the key signals in the bone marrow niche that actually help stem cell retain their potential, and do they get altered with aging?
I would like to know that. Sure enough, I'm pretty sure they're changing over age, but the exact point where stem cells are residing in their niches is actually what the field is after. So I can't really tell. It might be that there is interstitial fluid that is in contact with the serum or the plasma of an individual. It might be only that it is collagen that's getting more stiffened, and therefore it makes a difference, or ages like advanced glycation end products. They are all there. We know that, and maybe it's this kind of combination of aging factors that are changing, that are game changers.
I think another question that would be interesting would also be, could you comment on possible impact of hormonal changes such as estrogen or testosterone levels? Does that have an impact on osteogenesis during aging?
Sure. This is known. So estrogen is extremely important for osteogenesis as such, as a process, and it might be the case that if there is a big loss, so to say, of estrogen, and we know that this occurs during menopause, also changes something. But what we never found is a difference between female-derived and male-derived mesenchymal stem cells. We didn't see that, although you might consider female MSCs being more exposed to estrogen. So in this sense, we couldn't find a difference. But sure enough, for osteogenesis as such, this is well known. Yeah.
So there is estrogen signaling in males too. Is it clear that that has any role in bone and with aging in males?
Brian, I think it's well established that you need estrogen in order to form bones, and that's maybe as a base level for any type of signaling which you need to form bone, true for males and females.
Just a follow-up on that question. Did you take into consideration like the estrogen or the hormonal changes when you were trying to generate these 3D structures or organoids?
Not yet.
Okay. All right, maybe we move on to the more technical questions. So I think a lot of people have not really worked with 3D systems, so it's still quite a niche work. A lot of the questions are interested in the technique that you used. So one of the questions is, there are studies that show MSCs experience contact inhibition, whereas in your equipment, you are able to allow the MSCs to continue growing despite the contact, right? But based on your experience, do you find the MSCs continue to proliferate as MSCs, or are they prone to differentiate to other cell lineage if they are too confluent?
What we tried, and we are still into that we are taking these cells as they grow, and extract or isolate MSC-like cells and do all these kind of assays, which I showed you in the first part of my lecture. We are into that, and we think we have good reasons to believe that it's kind of in vivo-like, so that these cells are not turning into any kind of strange cell, like mouse cells can do, undergoing crisis, and then you end up with something like heavily proliferating cells. So they tend to be stable human cells. And sure enough, they tend to be diverse, as we have been able to show in the aggregates, that they self-organize from bone, they form different stromal compartments, and they also have a lot of proliferative cells around, which we don't know whether they are stem cell-like or just proliferating as they proliferate due to the open space which is around.
I think another technical question that was asked, did you alter the oxygen levels in the 3D system as well to ensure that they are near physiological levels or more ambient levels?
We did a bit of experiments there and didn't see differences to that extent which we saw in 2D.
I think there was also another question about topographical cues, whether it was something that you considered putting in place in your chips.
What in my chips?
Topographical cues, because apparently they are able to induce different types of MSC differentiation. So have you considered using this type of cues into your chips as well?
Yeah, I told you, what we are into is putting hydroxyapatite in, which we mix with PEG and try to get a mimicry of something like bony tissue. We have the cellulose hydrate, which might be able to offer a more sticky surface. So we are trying to manipulate that in this sense, but it's too early to tell whether we can succeed in forming something like a very, very artificial niche or bone marrow surrogate. But this is something that interests us a lot.
Thank you for that. I think there were also some questions on would there be a possibility if you were to inject exogenous MSCs to older individuals? Do you think there will be a rejuvenation, so more therapeutic implications?
It depends on what you think injection of MSCs is. 20 years ago, when the MSCs were kind of a fashionable cell, thinking that these can be used for this type of interventions, they ended up being dying, and they found an effect, and this was actually then thought to be something like an effect deriving. This is what people now tend to say, from extracellular vesicles that have been derived from mesenchymal stromal cells, and those extracellular vesicles at that time were thought to be immune modulating. So it's more what they produce as a cell type, rather than the cell type on its own that effects or shows effects that are could be considered rejuvenating or changing in something in the tissue. There's a lot of studies going on. I'm not into that anymore, reading everything what the outcome is here, because then one needs to read, I don't know, 100 papers a day.
Yeah, I do studies on MSCs too, and it's just a bit crazy. I think I was thinking when you said that, do we know why they don't engraft? I actually don't know the answer to that question. You might expect them to go to sites of damage and replace damaged tissues, but that's not been observed, as you say. But I actually don't know why, or if we understand that.
Yeah, a recent paper was using this kind of trick of aggregation, and now using the very, very classical way of aggregation, namely turning these aggregates into cartilage tissue. And there, this group used, or it's a consortium, used the cartilage for filling as a filler for osseous defects, and they could show that now you have an endochondral regeneration type, and depending on what you do to the cells beforehand, I don't know the details so good anymore, you'd end up with a certain outcome. So sure enough, what I guess the field is now striving at is using aggregates and putting that together maybe with biomaterials. So jump starting again this type of tissue engineering in vivo as the scaffolds together with the cells, with the factors that have been cultured in vitro and then reimplanted. They not totally failed, but virtually totally failed. So this approach, I think, is not followed up anymore.
Sure. Do you have like one more question or so?
Okay, maybe this is a personal question because as a PhD student in Brian's lab, so we are big on drug repurposing. So have you or your team tested any of the popular geroprotective drugs on this bone marrow aging model? And what interesting results have you gleaned? Not
No,
yet.
we thought of, we had one project which was on olive oil.
Okay.
And there is one, I think, which I can share as an idea. One gadget we're just building that we have something like a cell separator, and what it separates, it's actually according to size. So we're sorting out the large cells, which we claim are the old cells, and the leftovers should be the young cells. And preliminary results tell us that this actually should work. And I don't tell you how we separate the cells. It's a very, very simple device. You can just do that. It's pretty fast. It's extremely simple. And the only thing I can tell you, the only lab that can build that is us.
Okay, then maybe we have to collaborate on that. Thank you so much.
You're welcome.
Thanks a lot, Gunter. And by the way, as many of you who watch this show know, we have a healthy longevity talent incubator, and it lasts for a couple of weeks. We bring a lot of young students in of various shapes and colors, and teach them about healthy longevity. This is everything from MDs to basic scientists to business people. And Gunter is one of the faculty that was willing to come here for 2 weeks and participate in this course. I really want to thank you for that. I think that the young people really benefit, and I know it's a lot of work. And so thanks a lot for doing that, Gunter.
Thank you for invitation.
So I want to remind participants to use the chat function, and the panelists and all attendees option leave feedback and comments on the show. We will be back on a very momentous day, 4th of July, which is US Independence Day and the British elections. But if you don't have anything else to do, you can come on and watch our Healthy Longevity webinar with Professor Guo Longhua, an assistant professor at University of Michigan. I talked to him a few months ago, and he's doing something that I think will be interesting to you, studying regeneration and global age reversal in long-lived planarians. So a species we don't talk about very often. So be sure and watch that show. You've seen enough fireworks, and I'm not sure we really want anybody to win the British elections. So try the webinar instead. And tonight I want to leave you with a 102-year-old who's a champion sprinter. Thanks for joining us.
Catch him if you can. This 102-year-old athlete is proving that when it comes to competition, age ain't nothing but a number. This Thai man is his country's oldest sprinter. He recently competed at the 26th Thailand Master Athletes Championship, where older athletes compete in activities like the 100-meter sprint, long jumps, and the javelin throw. He gave his all during his events, and it paid off. The Century Club member completed the 100-meter run in 28.7 seconds, a new record in his age group, and he left the games draped in gold medals. Leading up to the games, he and his 70-year-old daughter/trainer train twice a day to get competition ready. His daughter shares that not only is her dad in good physical health, but he's also mentally stronger. She also says that before he began competing, his hands trembled. They've now stopped. The former teacher has been competing for the last 4 years, and he said the best part is making new friends. He said it also improves his appetite too. For Inside Edition Digital, I'm Andrea Swindle.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.